Good afternoon, everybody. Am I audible? Could you repeat? Am I audible? Okay. I'll just wait another 1 or 2 minutes until people join because we don't want half of them to miss the beginning. Please tell me if my screen is visible. Great, thanks. So, I think there's enough strength we can begin the class. So, good evening to all of you who have joined this live session. And this is the first session for this course, Bioinformatics, Algorithms and Applications. And I believe there are three other sessions which will be conducted, and there will be a total of 4 sessions every week for this particular course. And my other colleagues or other people will be taking up the same live interaction session, probably with a different content or different kinds of explanation. So, I'd like to know how many of you are at which stages of your career or educational career, either in undergraduate or postgraduate. You can always drop in inside, I mean, your replies in the chat box. And I hope this will be an interactive session where you can ask me questions. So we'll be discussing about 10 questions each week. Thank you, Ishan. Akriti, Kavya, Sheikh, Ritika, okay. And some postgraduates also. All right. Great, amazing. So I guess most of you are from a biology-based background, biotech to be specific. All right. So each week, the plan is to look into about 10 questions. It might be plus or minus here and there from uh, the week's content necessarily. So not very away from what is being taught. And we can also do some exercises because this is going to be a 2 hour session, and I'm available here for you to ask questions and clear anything that you might have. So let's get back to the PPT. So this week, the practice questions cover the topics, concepts and importance of bioinformatics, complexities in biological systems, DNA sequence analysis, sequence-based parameters, and database categories. Let's get into the first question. In a database, the list of valid terms and their definitions is called. So I hadn't linked in my, and I have not numbered all the questions, I guess, but that shouldn't be a problem. So I'm giving you a minute to. Okay. GPT. 1 second. I'll just give me a minute to log in through my phone so I'm able to see the chat responses. Okay. So you can hear, right? Alright. Now let me know if you can see the presentation. Okay, thanks. So, what are your responses? Option A by most people, I think. Unfortunately, I lost the ability to use polls because we lost the license. So, I cannot take a poll of how many feel what as the answers. Yeah. A is right. Ontology is the list of valid terms and their definitions. And all the others are not this. It's schema is how the website or the database is arranged. Query is what you enter to ask or obtain a data. Interface is where it's the user interface for the website, right, or the database that lets you access the information in it. Therefore the right answer is ontology. So question 2: purines found in DNA and RNA are. What are purines? Again, let's take a minute to share the answers. I think you can go with one or two. Okay. Use the question number. Okay, most of you are answering B. I saw some. Ishan says A. Ishwar also says A. Okay, Ishan is changing. Prasad also feels A. All the others are feeling B. Kriti, yes, the recordings will be uploaded on YouTube and you will soon be given the link. Okay, so most of the answers that I see are here A or B. There's no C or D, thankfully. So, first, DNA and RNA have bases which are categorized into purines and pyrimidines, right? What are the purines? Adenine and guanine. And the pyrimidines are cytosine, uracil, and thymine, right? So this naturally brings you to the answer adenine and where else is guanine? But the combination fits only in B, right? You should not get confused with cytosine, uracil, thymine here because they are pyrimidines in the first place. So irrespective of whether it is DNA or RNA, the answer is adenine and guanine. Right? Uracil is only in RNA. Instead, you have thymine in DNA. So that clears it up. The right answer is B. Question 3: which of the following databases store the nucleic acid sequence information? Okay, so many D's. Okay, that's right. So protein data bank has the information of protein structure, so it's wrong. Nucleic acid database is there, but I don't think there's data bank. NA-seq data seq bank does not exist, so it is Gen bank which stores nucleic acid sequence information. Question 4. Which of the following statements is not correct? Is it option A, RNA contains adenine and guanine? B, DNA contains phosphate and deoxyribose sugar? C, RNA contains cytosine and uracil? And D, DNA contains cytosine and uracil. So what did we just see? Adenine and guanine are going to be present in both DNA and RNA, so this is right. Option B, DNA contains phosphate and deoxyribose sugar. Yes, that's true. RNA contains cytosine and uracil, which is also true. But DNA contains cytosine and thymine, not uracil. So this is the right answer. D will be incorrect. Yes, Mohit. So it is correct. Okay. Question 5. Okay. Very simple question. Yes, right. So UAG, and UGA, all three are stop codons, right? AUG is the only start codon which codes for methionine. Right answer. Question 6. Which of the following describe bioinformatics? Okay, some feel A, some feel B, and some A and B. Okay. Yes, it is MCQ and there's only one right answer. So what would you choose? Okay. Yes, as Mohit pointed out, you have to go with the most appropriate option compared to what are all the possible options. Okay. Although protein three-dimensional structure seems to attract a lot of people, it is not a complete bioinformatics thing, right? Although it is possible, you do have AlphaFold to help you with that, but there are other means to also arrive at the protein's three-dimensional structures experimentally. Sanger sequence is experimental, gel electrophoresis is another experimental or analytical tool, which does not involve bioinformatics. Therefore, all are wrong compared to molecular docking, which is purely bioinformatics. Question 7. Number of overlapping trinucleotides in for a DNA sequence of length 10 nucleotides is. You can either use the formula that Sir gave during his lectures, or if some of you have not gone over the lectures, let's say you have 2, 3, 5, 6, 7, 10. Okay, so this would be one. Number of trinucleotides, overlapping trinucleotides, so which means this is also allowed, 2, and this will be 3, 4, 5, 6, right, 7, and finally 8. So when you're talking about trinucleotides, you will be leaving one on either side of the sequence, right? So n minus 2 would give you the answer. I think that's a bad color. So n minus 2, so that would be 10 minus 2 is equal to 8 overlapping trinucleotide units, right? The answer is 8, as most of you got it right. Great. The GC content of the DNA sequence ACGCTGA is, okay. So what would you go for? Number of G or C divided by length of sequence into 100. For those of you who haven't arrived at the answer yet, so how many C and G's do you have? 1, 2, 3, 4, 5, 6. And how many are there in total? 1, 2, 3, 4, 5, 6, 8, 9, 10. So that will be 6 by 10 into 100, which is 60%. The answer is 60. Right? Okay, question 9. This is the simplest question you can get. So, number of hydrogen bonds between G and C is, great. So, what is the number of hydrogen bonds between A and T? Okay, all right. So, the answer is 3 for GC pairs. Which is the most important factor for a database? Is it availability, correctness of data, speed of connection, or retrieval of data? Again, you have to go with the most appropriate or the most significant, whichever option carries the greatest weightage. So, is it option A, availability at all times, the availability of data, and the speed of connection, how fast you can retrieve the data, and the retrieval of data, the ease with which you can find what you're looking for, or the correctness of data, how reliable the information is? So some have responded D, some have A and B. Okay, although all four are necessary for the creationing and for creating and maintaining or functioning of a database, it is the correctness of data which is the most important factor. Even if your website or database is always available and the connection is amazing, retrieval of data is very perfect, but the correctness of data or what you have tabulated or gathered is wrong, then there's no point in having the database in its first place, right? So the answer is correctness of data. This is 9, 10. Oh, okay. With that, I think this was really quick because all of you were very enthusiastic in answering. So if there's until now, if you have anything that you want to ask or clear amongst the questions that we discussed or anything else, you can ask. Otherwise, we can do some small exercises to keep ourselves busy for some more time. So I'll wait for 1 or 2 minutes until you type in the questions that you might have until now. I'm very sorry, I have no idea about how the final exam would be because I'm not part of the NPTEL team that creates the content or sets the questions for the assignment or conducts the exam. So I'm just here to, yeah, you can ask people who have taken NPTEL exams previously, or I can find out and let you know in the next class. Okay, how to calculate average hydrogen bond energy? So let's take a example question. Mhm. I think you had one question in. Is it there in current assignment? So if it's there, we won't use the same question, but we'll do something similar so that, or we could generate. It's not a big deal. It's just an example that we're going to do. I'm just randomly writing, so don't think these are real values or. So have I covered all TC is missing, right? Anyway, let's see what we miss here. So GC, CT, TA, AT. Okay. So I think this is enough for us to do this particular problem. So let's take uh, GC is minus 2 plus CT is minus 2.5 plus 45. One second. TA is minus 3. AT is again minus 3. Right? So what does this give you? 3 plus 3, 6, 8, 10.5. And I'm going to divide by n minus 1, right? Because I'm taking it in dinucleotide units, correct? So this will be minus 10.5 divided by 4. Oops. What is that? So the answer is minus 2.625. Is everybody clear with this? Let me just check if. Otherwise, I think the assignment is pretty easy. We could do another problem. Let's see. I hope all of you understood this example, correct? I mean, GCTAT1. Yeah, it was clear, right? Okay, so we could do one more, not hydrogen bond calculation. Let's say you have Kritika will stick to the topics of this week. Anyway, all those will be covered as we progress. Okay, because not everybody here is familiar with everything, so we'll just take it stage by stage. So find the complementary sequence and report in 5 prime to 3 prime format. So how would you do this? Okay, Yagesh is already answered. Let's check if he's right. So first, what do we have? A T A T C G A T C A G T, right? So can we conclude that this is right? So when complementary, you are anti-parallel, right? Therefore, when you write it backwards, 5 prime to 3 prime. So these are very simple questions, but you might make mistakes while choosing MCQs, where you have to be careful what you choose. You cannot jump in and see the option and tick. Instead, you have to wait and check what orientation the answer is given in. Right? T G A C T A G C T A T A. I think most of you got it right. So for Gaurav, the formula is nothing. So whatever sequence that you have, right? Let's say A T G C C T G C G or C T G. Okay. Now the number of nucleotides here are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So how many doublets will you be able to make? 1, 2, 3, 4, 5, 6, 7, and 8. Have I missed something? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 1, 2. Oh, okay. GC I have missed. So there are a total of 9. Right? Whenever you're trying to make couples out of a particular sequence which are overlapping, right? You will end up with n minus 1 binucleotide segments or units. So that goes into your denominator because those are the number of units that you are calculating for. And energy, whatever I gave were random numbers, and in your question it will be you will always have a table of information where the interaction between two nucleotides are given, or two base pairs, not even two nucleotide base pairs are given. Let's say T, G, or C, T. Now, whether it is AT or TA, you will refer to the value given against this AT. Whether it is GT or TG, you will refer to the value given against TG. So, like that, you sum up each of these interaction energies and divide by the number of units. If it is triplets, it would be n minus 2. If you're talking about doublets, you will do n minus 1. How do you know whether to choose triplets or doublets? Based on the information that is given to you in the question or the tabular form. Okay, I hope that helps. Okay, somebody has asked for complementary sequence. So 5 prime, if you have a sequence, so G always pairs with C, A always pairs with T. T always goes with A, G always goes with C. C again will go with G. So this is called complementarity of sequences. This is for you, Sandhya. So where particular nucleotide base always pairs with another. Uh, so we started our discussions on purines and pyrimidines, right? Adenine and guanine are purines which have, not drawing the exact structure, but they have shapes, or the structure is such that they have two rings. And pyrimidines, cytosine, thymine, and uracil, have a single ring. So basically, pyrimidines, although the name is big, they are smaller molecules, whereas purines are bigger. So when you're trying to maintain a DNA, which is like a ladder where each base pair is stacking over one another, if you have two tiny things and two huge rings in the middle, you will not have a ladder, it'll rather be squiggly like this, right? But the DNA is never like that. It's actually a straight piece of or a parallel piece of information which then winds on itself to look helical like this. So, in order for the distance to be maintained properly between the two phosphodiester or sugar phosphate backbone, essentially, or the two strands of the DNA, you need to always have a purine marrying a pyrimidine. Or the purine should always pair with pyrimidine, or pyrimidine should always pair with purine. So, people have seen by experimental methods, and over the years, it has been found that adenine always pairs with thymine, or uracil in case of RNA, and glycine always, I'm sorry, guanine always pairs with cytosine. Okay, and vice versa. Cytosine will always pair with glycine, and thymine will always, or uracil will always pair with adenine. And while these have two hydrogen bonds between them, pairing between G and C will have three hydrogen bonds between them. That is why you always say or go with a sequence strength or rigidity based on the GC content, right? So, with this background, this is how complementary base pairing happens. And when you're asked to write a sequence, or what is the complementary sequence of GACTG, it is a general good practice to start with the 5 prime end, right? So you should go with C A G T C, because that's the one, because you always start a sequence from its 5 prime end, and 3 prime is generally the tail, the head. I hope that was clear. Okay, so let's do another question where we calculate the number of. So if this is the sequence, what is the GC percentage and 80 percentage? This is question A. B. C would be, what is the let's see. If this were an mRNA, how many reading frames are possible? Okay, Kavya has and Anas have replied. Shall we check their answers? So we're going to with. I hope the screen is visible, or should I go in presentation mode? Do you want me to? So let's first do GC. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 19. Nice. So that you have to waste some time in calculating. It's not as straightforward as 6 by 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So 11 GC and 1, 2, 3, 4, 5, 6, 7, 8. Just to confirm that I haven't left any ACs. So GC would be 11 by 19 and 80 would be 8 by 19, or simply 100 minus GC percentage, or 100 minus 80 percentage should give you the other one, right? So 11 divided by 57.89 for GC and 42.1. Doesn't add up, does it? One more one there. Okay, so A was 57.89, B was 42.11, and many of you have answered for C. Great, 6 reading frames. And for those of you who have not paid attention to lectures, how do you get the other? So, as codons or triplets, you have to go with 3 reading frames on the, assuming this is 5 prime to 3 prime end. On the other side, you will have a complementary sequence with yet another sequence. Like I'm leaving. One second. There's no more. 1 second, please hold. There's no more in the lab, so I'm leaving. You can lock it. I can lock it. Let's go. I'll lock. Okay. Uh, where were we? Yes. On the complementary side, you will have G, A, C, G, 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 right? So again, you can start with G, or you can start taking codons from A, or you can move from C, G, G. But keep in mind that this is just an abstract place, and you should always start with wherever the open reading frame starts, right? Only when if you have methionine, you can go continue and worry about codons. Otherwise, if there's no AUG, there's no point in making codons and worry. But just to get the idea that you will have 6 different frames. If this is 1, 2, and 3, this will be -1, -2, and -3, based on which is the template and which is the complement, right? So, what else? I'm thinking of giving one more. Uh, okay, so I had looked up something for you to check out. So I want you to go to DDBJ website now, database, and search for human salivary mileage, just to save time and all of you do the same exercise. Or you could search, I think I had it. It's there. Yum 1867.1. I think, I'm not sure, but I had it here. Yes, 18671. There's no point in between. So I want you to load this find the total number of base pairs and what is the number of, let's say 80%. Okay, Shreya wants the reading frames concept again. So while I explain that, the others can go ahead and find this. But if I use another slide, you won't be able to see this information, I guess. Right? So let's just on the concept of reading frames again. Okay. So whenever you have a sequence, let's take G T C G C T A T G U G. Oops. C G G C C T G A. Okay. So Shreya, I hope you're aware of the concept where, assuming you have some start codon before this, you could always uh, find from the table of amino acids, right? Like how you know AUG codes for methionine, or I think UGG codes for tryptophan. I'm not sure. Look it up. But there's this table where you have all the 64 possibilities because you're having 4Q, right? You can pair with any of them. AUG, I mean, you will not be using T, instead ACUG. So when you pair these four bases in all possible combinations, you will get 64 possible combinations, right? Thank you, Shreya. So you can have codons which code for a particular amino acids, and that's how you take the protein sequence, predict the protein sequence from a nucleic acid sequence. Now, you have a nucleic acid sequence, and you want to predict what is the possible protein sequence, amino acid sequence that you can get from it. How would you know where to start? So if I start from GTC, I would assume it's going to be you and you're going to transcribe this. You will have, let's say, I'm just going with, since I don't by heart remember what these amino acids are, I'm just going to go with A, B, C, D. Okay. But if I start from here, it will no longer be A, B, C, right? I might have something like E or F or again C, I don't know, just for argument's sake. And if I start from C instead, I'll get something else. It will be CGC followed by TAT followed by GCG. So you could either go as GCT, ATG. This is first possibility. The second one is TCG with G left behind, and you continue with CTA and TGC. And the last one would be GT and CGC, TAT, and so on. So these are three reading frames in itself, 1, 2, and 3. And to complicate furthermore, or it is still the same sequence where, or the same gene, which in its complementary sequence can give rise to 1, 2, and 3 reading frames, and these are called -1, -2, and -3. So therefore, you get 6 reading frames in total. Great, nice. So Anas, Prashant, and Afra have responded. It is true. Okay. Yes, let's go check if their answers are right. So I'm going to click into M1867 671 18671. So the number of base pairs are, I can't draw here, 1071 base pairs. And next, where do I find the ATGC content? Here. So first I'm going to write down what is the total number of base pairs as 1071, and for AT I'm going to note down what A and T are, 294 and 358. 294, 358. So what would you do? 294 plus 358 divided by 1071 into 100. That would be 294 plus 358 divided by 1071, which is 60.87. That's right. Great. All right. So I think I'm done with the questions or exercises that I prepared for today. I also had one where you can go and search, look into the Emboss website and find the same complementary sequence or transcribe something, but you could always do it in your free time. So if there's anything else, I would gladly help you with it. So I'll wait for another 2 3 minutes. Where is CDS? Okay. Oh, I'm not. So where did you find this? Okay. I think somebody came up with the answer. Right. I mean, these are things that are very user friendly and you can always find it yourself. Okay, anything else? Sandhya, anyway, the PPT will be shared. Both the recording and the content will be shared. I think there's one more question from Prashant. What is the difference between version number of a sequence and the gene index? Um, Again, Prashant, I think I have to get back to you on that. There's a list of an ontology of what each of them mean, the version number and the sequence, but I don't remember it off my head. All right then. So hope to see you all next weekend, Friday, same time. Thank you. Okay, I don't think that's possible. I would, of course, like to. When the WhatsApp or Telegram group is not possible. All right. Thank you all for joining. I'll be closing down the session. Yes, Ritika, unless you have something else to ask. All right. Thank you.